Enjoy the event and have a great festival.
So, good afternoon. Thank you to Sophia, our AI-generated avatar, and thank you to you all, to this wide audience, and thank you and welcome Professor Wieland Huttner. So, today we have a lecture devoted to development and evolution of the human brain, so a very fascinating issue. I would like to emphasize that Professor Huttner is Director Emeritus and the co-founder of the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany. He's a biochemist and neuroscientist, and he's really, let me say, a pioneer in the research devoted to, let's say, the most mysterious organ of our cells, the brain. And as a matter of fact, his research primarily focused on the cellular and molecular mechanisms behind the human brain's unique transformation and growth. So, thanks to his studies, he has shed light on how our brain has evolved to become vastly different from those of other species. So, first of all, primates, but also the other hominins, so from the Australopithecus up to the Neanderthal. And in particular, his contribution lies on the research of the neocortex, the part of the brain responsible for language and for the symbolic thought. So today we'll undergo a really fascinating story involving cells, neurons, proteins, and genes, and also all the networks that make altogether our brain such a powerful, earth-surprising machine. So today we'll start a journey in our deep past, and we will see what happened in that long span of time, while opening many new perspectives and arising many questions. And I would say that the main one is, what makes us humans? So, Professor Huttner, thank you very much for joining us, and we are ready to listen to your lecture. Thank you.
Okay, so good afternoon. Thank you very much for your kind and professional introduction. And I would like to express my sincere thanks to Professor Zeviani, who invited me to give this lecture, which I consider to be a great honor. And of course, it's an incredible experience for me to give a lecture to this fantastic audience in this even more fantastic setting. Unbelievable. It's really a great, I can only repeat myself, it's a great honor. I also learned that my black slides have some green vertical lines. Hopefully, not only the black ones and not the white ones. Okay, let's get started. So development and evolution of the human brain is a wide topic with many, many different questions. And today, I will address two questions. And no green lines, good. The first question is, how did our brain become so large in the course of human evolution? And second, were there differences in brain development between us and Neanderthals, our closest relative? These are all questions that, as you said in your introduction, directly address the question, what makes us human? And the comparison to Neanderthals, we do because I belong to those people who think that Neanderthals were not of the same cognitive abilities as we are. We flew to the moon, and I make this bold statement that I don't think Neanderthals would have flown to the moon even if they lived now. So here you see several mammalian brains. The size of the brain, 1 centimeter scale bar, is shown here. And you will see numbers here in these branches. And these numbers indicate the million years where the lineages segregated. So in other words, 7 million years ago, the lineage that lead either to the chimpanzee or to us, segregated. And another thing you notice on this slide is that certain brains are not folded, they are smooth, like the mouse, but other brains, including ours, are highly folded. The purpose is to fit more cortical, cerebral cortex into the skull, the volume of which in mammals is limited because of the birth canal, as you can all imagine. So addressing the issue, how did brains in evolution get big, and especially what is the cause for the tripling in size from the chimpanzee brain to us, is a fundamental question. And we know this because there are unfortunate human individuals which, due to mutations in certain key genes, are born, or can be born. What's the problem? It's fine like this now, huh? Are born with the size of a chimpanzee, which is a third of normal. These people are intellectually disabled, which makes the point that brain size is one factor for our cognitive abilities. On the other hand, these people are, although their brain is only the size of a chimpanzee, a third of us, they're much smarter than chimpanzees, which makes the other point, I'm sorry for this, I don't know why it's flickering, which makes the other point that brain size is only one factor for cognitive function, not the only one. So our goal was to identify genes, or a gene, Is it better now?
It's flickering. I don't know why. Prego? Prima non faccio... no, ma è il computer. May I try to disconnect?
Yes, sure. I'm sorry for this. No, it's different. Now it's better. It's a different shape. Okay, let's hope it doesn't start to flicker. Okay, so the goal we had was to identify genes, or hopefully a gene, that could explain this tripling in size of our brain compared to the chimpanzee. Now, when during development a brain gets bigger, the stem cells of the brain generate more nerve cells, or also called neurons. And today I will focus on the cerebral cortex, which is this structure here, this convoluted structure, and notably the neocortex, which is essentially the whole thing here that you look at, which is the evolutionary youngest part of the human brain. Now, I mentioned that stem cells are key, and I will give you a little bit of an introduction to stem cells. What you see here is a section through a mouse embryonic brain. And you see blue little structures, which are nuclei of cells, specifically of stem cells. And then you see green little ellipses, which are nuclei of newborn neurons that are born from these stem cells. It's still flickering or not? Thank you. Okay, so the stem cells of the developing brain in mouse and in human, it's the same, fall into two classes. We have a class of stem cells that is right near the ventricle, which is the apical side of this epithelium. in a structure in a germinal zone called the ventricular zone, and these progenitors, or stem cells, do not make neurons. They make a second class of progenitors that are a little bit more in the basal direction, which we call basal progenitors, which reside in the subventricular zone. And it is these stem cells, the basal progenitors, which make the neurons. Now, the basal progenitors are key for a process called neocortex expansion, which in lay terms is how our brain got bigger. And I will illustrate to you why that is, why the basal progenitors have such a key role. The apical progenitors can divide only at a very, very thin area here at the ventricle, which is not good if you have a limited space and you want to maximize cell division. In contrast, the basal progenitors have much more space in the subventricular zone to divide. So they can make many more cells, many more cell divisions, many more neurons. And this is mouse. In human, the area of the red area here versus the blue area here is 100 to 1. So a clear indication why the basal progenitors, which are amplified in human, are so important for the expansion of the neocortex. So with this in mind, we initiated a search about 10 years ago for human-specific genes, so genes that occur in humans but not in chimpanzees, our nearest living relative, genes that are preferentially expressed in the stem cells of the fetal human neocortex, because that's where we want to study brain expansion, and genes that can increase the basal progenitor proliferation and abundance, so the ability of the basal progenitors to divide. And when I say we, I talk about a fantastic PhD student from Italy, from Milano, Marta Florio, absolutely outstanding, and she was supported by 4 other people in my lab. Now what Marta succeeded in identifying is a human-specific gene with a horrible name called RGAP11B, or simple for this afternoon, 11B. And that gene is, as it turns out, to be really absolutely fascinating. When she found that, the first thing we checked is whether that gene would do what was our prerequisite, that it amplifies basal progenitors, that it makes more of them. And so we used the embryonic mouse neocortex as a test system. And when we express this human-specific gene in the embryonic mouse neocortex, we see a massive increase in basal progenitors. So the gene does what was our prerequisite it should do. So it's a human-specific gene preferentially expressed in cortical stem cells. The other thing that made this gene really, really interesting is how it arose. So the gene arose as a product of a human-specific partial duplication of a widespread gene called RGAP11A. And that is work not by us, but by Evan Eichler in the US, who has discovered that gene. And so we have 2 genes, RGAP11A and RGAP11B, and they encode the following proteins. So these stretches that you see is meant to be a protein. So 11A is a very long protein with more than 1,000 amino acids. And 11B is in essence a copy of the N-terminal or the left 1/4 of 11A. But there is this green part in 11B that does not exist in 11A. This green part consists of a stretch of 47 amino acids. And this stretch of 47 amino acids is unique, only occurs in humans, and it is essential to increase basal progenitors. And I'll show you this later. The other thing that fascinated us was the time point when this gene arose. This gene arose about the duplication of the gene, occurred about 5 million years ago, which is after the lineage that leads to our nearest living relative, the chimpanzee, segregated from the lineage leading to us modern humans, which was 7 million years ago, as I showed on my first slide. But it, of course, 5 million years is much, much earlier than the segregation of the lineage that leads to Neanderthals and to the Denisovan hominin, which are the closest relatives, although no longer present on this planet. This segregation here took place about 500,000 years ago. So we had a gene then that was expressed in the relevant stem cells, and it was only found, or is only found in brains that are big, 1.4 liters, 1.3 liters. So that led, of course, to the question, can the evolutionary increase in our brain during evolution, can that be due to RGAP11B? And we have a problem with that question. Because I said the duplication took place 5 million years ago. But 5 million years ago, hominin brains were small. The big surge in increase starts about 2 million years ago. So it seems that this gene is too old to do such an important thing as increasing brain size. But we have a solution to this problem. And that solution has to do with this green piece here. Because the key question is, how did this C-terminal sequence, so at the end of 11B, that is unique, occurs only in humans, and is essential to make basal progenitors, how did that sequence, this green bit, how did it arise? And now it's becoming a bit complicated. I will not go into details here, but just summarize how it arises, because that's stunning. Sometime after the gene arose, so sometimes after 5 million years ago, there occurred a point mutation. A single base, a single nucleotide, changed from a C to a G. You know there are 4 letters, A, T, C, G, and 1 C changed to a G in the 11B gene. This point mutation leads via a complex process that I won't go into called RNA splicing. This point mutation leads to the human-specific C-terminal amino acid sequence of the 11B protein. And that human-specific sequence is essential, as I said, for increasing basal progenitors. In other words, the ability of 11B to do in evolution what we think it does, which is to increase basal progenitors, is not the result of the duplication of the gene as such, but of a point mutation that occurred later. Let me summarize this. The gene, as it arose 5 million years ago, did nothing on brain size, because it lacked the critical green C-terminal sequence. Then some point later, we do not know exactly when, but an attractive thought is maybe 2 million years ago or 1.5 million years ago, there is this point mutation in the gene, which gives rise to this human-specific green sequence, which in turn underlies the ability of basal progenitors of RGAP11B to increase basal progenitors and to enhance the neocortex. And interestingly, this point mutation exists not only in us, you, in all of you actually, but also in Neanderthals. And the brain of Neanderthals was at least as large, slightly larger actually, than our brain is. So that leads then to the question whether this gene can actually expand a primate neocortex. We are primates, as you know. And so we tested that by making transgenic monkeys. This is a so-called common marmoset, a New World monkey, a tiny little creature, very cute monkeys, very nosy, very intelligent. And we expressed this human-specific gene in these monkeys at the level of their fetal stage. And this is a collaboration with Hideyuki Okano and Erika Sasaki in Japan, who pioneered the transgenic marmoset technology. And it's work done by another fantastic postdoctoral fellow in the lab, Michael Heide, who went to Japan to learn this technique. What you see here on the left is a normal wild-type fetal marmoset brain at day 102, which is 2/3 through gestation. And you see this brain is smooth, nothing special about it. Note the scale bar, because what I will show you in a second here is what happens when you add to this fetal marmoset the human-specific gene RGAP11B. Same scale bar. This is what happens. The brain gets bigger and it begins to fold, which is a characteristic of our brain, of course. So 11B can expand the cerebral cortex of a primate and induce its folding. How does this gene achieve its effects? That's another fantastic postdoc from Japan, Takashi Namba, who did this work that you will hear now about. And he started out by saying, well, where in a cell does this gene operate, or does the protein rather operate? And when you do an immunostaining for this protein, you see these, I'm German so I can say this, these sausage-like structures. And when you know this as a cell biologist, you immediately know what these structures are. These are the mitochondria, the powerhouses of a cell. And here is the double stain to prove it. So the localization of 11B protein in mitochondria is essential for its ability to increase basal progenitors. What does it do in mitochondria? It's a collaboration with Christos Yiannopoulos in Budapest. So 11B is in the mitochondria and it binds to another membrane protein of mitochondria, but via its green human-specific C-terminal sequence, it inhibits a structure that is referred to as the mitochondrial permeability transition pore. Never mind the details. The end result of that inhibition is that a metabolic pathway called glutaminolysis is stimulated. Glutaminolysis is a pathway where the amino acid glutamine is converted to glutamate, which you eat in good Chinese food, which then in turn is converted to alpha-ketoglutarate, an intermediate in the citric acid cycle or TCA cycle or Krebs cycle. When we realized that RGAP11B protein in its mitochondrial function would stimulate glutaminolysis, we became very excited, because glutaminolysis is a hallmark of cancer metabolism. So our human brain is so big because the stem cells of the developing brain adopt a cancer-like metabolism as long as the gene 11B is active. And hence the stem cells of the developing brain divide more frequently, which is typical for cancer cells, and thus generate more neurons. And that's it. So now the question was, does the neocortex expansion by this gene result in improved cognitive performance? It's work by another fantastic postdoc, Lei Xing, and Mikhail Sarov from Bulgaria, who runs our genome engineering facility. And it's a collaboration with very dear colleagues in Prague and the Czech Republic, Radek Sedlacek. So what we made was a so-called transgenic mouse, where we inserted this human-specific gene so that the mouse would now make this human-specific protein. These mice have a slightly larger neocortex. That's not a surprise. You will see this here. And when we quantify that, there is a significant increase. It's not a big increase, but this is because the way we expressed the human-specific gene was not super strong. It was a very moderate, mild expression. But these mice also have more neurons, as shown here. And now the question is, are these mice, which make a human-specific gene, protein, are they smarter? So we sent the mice to Prague, to our colleagues in Prague, who are world experts in measuring mouse behavior. And they subjected these transgenic mice to a battery of tests. I will just show you the result of one test, which interestingly is called IntelliCage. It tests the cognitive abilities of mice. So when we do this and let our mouse be tested in Prague in these IntelliCage tests, what we see is that in contrast to the wild-type mice, which take a while to learn things and make errors while doing so, the error rate of the 11B mice is half. So they learn much faster. They are simply smarter, which means that they show improved cognitive performance. So neocortex expansion by this human-specific gene does result in improved cognitive performance. And this is shown on this cover where we published this in 2021, where you have a wild-type mouse and the transgenic mouse with a bigger brain, which beats the wild-type mouse in a memory game. I had told you that this human-specific gene with a crucial point mutation also exists in Neanderthals, which is consistent with the fact that the brains were super big. With this, I would like to end the story on 11B and the gene that makes the brain big, but now go beyond size, because I had said in my first slide that brain size is one factor for cognition, but not the only one. And I would like to ask the question, were there differences in neocortex development between modern humans and our closest relative, the Neanderthals? And I will tell you one story this afternoon. And this story is a collaboration with my friend Svante Pääbo, who got the Nobel Prize in 2022 because of his success in sequencing the entire genome of Neanderthals. And here you see how he gets the Nobel Prize from the King of Sweden. So the work was done by another outstanding postdoc in my group from Costa Rica, Felipe Mora Bermudez. And what Felipe did was he asked the question, were there differences in the behavior of brain stem cells between us and Neanderthals? And the story started about 8 years ago when Felipe studied a model system called cerebral organoids. These are, if you wish, mini brains, a few millimeters big, which are grown from pluripotent stem cells, and they make little ventricle-like, brain-like structures. So these little ventricles, they have apical progenitors, they have basal progenitors, and they have neurons. So Felipe said, let's look at the mitosis, the cell division of these apical progenitors. The power of these organoids is that you can make them not only from human pluripotent stem cells, but also from apes, which are highly protected. So these are actually chimpanzee organoids, which look at this level very similar to human organoids, but this technology of organoids allows us to compare apes, which are highly protected, as I said, with human brain. So Felipe imaged this division, the mitosis of these progenitors, and I will show you now a movie, assuming that you have not yet seen how a human stem cell divides, and certainly not how a chimpanzee brain stem cell divides. So you will see how this nucleus, which is a bit pixely because we avoid phototoxicity, and this chimpanzee nucleus move down here to the ventricular surface, round up in pro-metaphase, in green, make a so-called metaphase plate, and then go into a part of mitosis that's called anaphase, which is when the chromosomes, which are still together, go apart to end up in the two daughter cells. So here is the movie. So you see how these nuclei move down, round up in pro-metaphase. Now in yellow they make a metaphase plate, and now watch the chimp, boom, anaphase. And human, boom, anaphase, but later. So we have more time in this anaphase part of mitosis than the great apes. That's true for the chimp, and that's true for the orangutan. Now you would say, come on, give me a break. These 4 minutes or 3 minutes longer, that cannot be important, right? I mean, cell cycle is hours. How can 3 minutes make a difference? But they do. And I will explain to you why. So during metaphase, and specifically during metaphase, the correct alignment of the mitotic spindle with the chromosomes is checked. As you know, when a cell divides, ideally the 2 daughter cells should get an equal number of chromosomes of the genome. But there is a checking system. And when this checking works, then the spindle is completely symmetric and the equal number of chromosomes end up in the 2 daughter cells. But when this checking works incompletely, you have chromosomes that are no longer attached to the spindle and they don't know where to go. They can go into the right daughter cell or into the wrong daughter cell. So given the fact that the chimpanzee metaphase is shorter than human, Felipe said, well, maybe the checking in the chimp doesn't work as well as in us. Maybe the chimp has more lagging chromosomes. And that's what he found. The chimpanzee has twice the number of lagging chromosomes. Now, which proteins do this checking? There are 2 proteins, again, large proteins with 2,000 amino acids here and almost 1,000 here, called KNL1 and KIF18A. Details don't matter. But the interesting thing is, in these 2 proteins, between the chimpanzee and us, there are just 3 amino acids that are different. 2 in KNL1, here, and 1 in KIF18A. But what blew our mind, literally, was when we realized that Neanderthals have the same amino acids as chimpanzees and as mice. In other words, we and Neanderthals are strikingly different in these 2 proteins that do the checking of the chromosome distribution. So because the ancestral amino acids are in Neanderthals, in chimpanzees, and in mice, we made use of mice and said, let us humanize mice. So we replaced in mice, by genome engineering, in these 2 proteins, just the 3 amino acids to the human type, the modern human type, and asked, would that do anything? And the amazing thing was, compared to the wild-type mouse, which is Neanderthal-like, when we humanize the mice in just 3 amino acids and 2 proteins, the mistake rate, the lagging chromosomes, go down to the level of us. Just humanizing 3 amino acids makes these mice, in this process, behave like human cells. Now, the implication of these data is that brain stem cells of Neanderthals made more chromosome distribution errors than brain stem cells of modern humans. So the answer to the second question I asked, the first question was, can we identify a gene? And we did. And the answer to the second question is there were differences in brain development between us and Neanderthals, our closest relative. And with that, I would like to end to show you our Max Planck Institute in Dresden and to show you a few slides of Dresden, because Trieste is, of course, a beautiful city, super. But Dresden is not so bad either. So here you see Dresden at night with the Semper Opera, which is very similar actually to this fantastic opera here. Here is the opera at higher magnification, if you wish. Beautiful building like this one. And this is the reconstructed church, which, as you will know, was destroyed at the end of World War II. The dark stones are part of the old church. You see a lot of this here, but there are individual dark stones here, and they are put into place where they were in the old church based on the stone mason signs. So that's how they could be placed in the proper position. And with this, I would like to thank the funding organization and to thank you for your attention. Thank you.
So thank you, Professor, for this wonderful and interesting lecture. I'm sure that you open up many more questions and possible perspectives. So I'm wondering if in the audience there's someone who would like to ask a question. Do we have... we'll have a microphone probably coming. Can we have a microphone?
You shout and I repeat it.
Okay.
Thank you for the wonderful lecture. And I was wondering whether there are implications on the epigenetics. So the passing of epigenetic information from one generation to the other on this last time.
You mean epigenetics, right? Yeah. So the gentleman is saying, is there a role of epigenetics in passing on this information to the next generation? Epigenetics is a field, a relatively new field in science, where one realizes that the DNA sequence is not the only thing that determines what happens in a cell, to put it in very simple terms. And yes, there is very increasing evidence. Actually, one of my postdocs, Mareike Albert, is a world expert on epigenetics of brain development. And this is ongoing research by her, but there's clear evidence in line with what you suggest with your question, that epigenetics and epigenetic information is also passed on to the next generation.
Thank you. Okay, we have another question. Can you try to shout while waiting for the mic? Thank you.
Sorry, can you raise your hand where you are?
Can you...
Okay, I see you. I see you. No, I see you. Okay. Can we find what? Yes, of course, the mutation of the gene, you mean the point mutation? Yes, this is present in all humans. So basically every single human has the G, all of you have the G. There are 8 out of 800,000 human beings that have it only on one of the 2 alleles. That's very rare. But there are no human beings which have no G and only the C. And this mutation is passed on. It passed through the human population sometime between 5 million years and 2 million years, which is a lot of generations, right? If you assume a generation time of humans of 25 years, you have lots of generations to allow it to pass this mutation through the entire human population.
Okay, we have a question here. Thank you.
So I try to repeat the question. So this gentleman says, well, this mutation happened relatively recent. We don't know exactly when, 2 million years ago or something like that. And it had a big impact on why we are the way we are and why we have a big brain, or a 3 times bigger brain than a chimp and a 50% bigger brain than Homo erectus. Yes, that is true. And this mutation almost certainly is stabilized in the population. We don't have any proof of this yet, but because of positive selection, it's not you don't need much imagination to assume that when you have that mutation and your brain grows, that this is a selection advantage over hominins that don't have that mutation. So it's no surprise that it's spread through the entire human population. I don't know whether this answers your question. Okay. Yeah, I mean, of course, brain size is sets the stage for our cognitive and technological developments, but of course this mutation is not directly responsible why we flew to the moon. I mean, that is of course the overall cognitive function of our brain, which involves lots of genes, I'm sure. We don't know them yet, but genes that make the wiring of our nerve cells proper, that affect synaptic function, and so on and so on. But the stage for all of this is, in the first place, you need a sufficient number of neurons, and that is what that gene does.
Okay, we have there, okay, please.
Raise the hand, yeah, okay, good. I'm not sure I get the question. The question is how we found these for the second story, how we found these proteins. Well, I can. So, let me say how we found them. We as Svante Pääbo and myself together. So there are about 100 proteins between Neanderthals and modern humans where there are a small number of amino acid changes. So we went through the list of these proteins and said which of these could do the checking of the spindle chromosome contact. And that's how we ended up finding them. So we searched the list of 100 proteins for possible functions in chromosome distribution, and that's how we found them.
Thank you, Wieland. This was a wonderful presentation, of course. So my question is, as you well know, Neanderthal and sapiens inbred themselves only in Europe and in Western Asia. So I wonder whether some traits or genetic traits of Neanderthal were inherited by Europeans but not other populations, and whether this makes a difference in terms of health or somatic traits or else.
Okay, so that's a very, very pertinent question. Yes, so Neanderthals left Africa before us. And when we left Africa, we interbred, we married, no, not married necessarily, but we got kids together with Neanderthals. And the end result is that here the people from Europe, all of you, carry about 2 to 3% Neanderthal genes in you. So people always say Neanderthals died out. It's not quite true. They ended up in us because our population was much larger than that of Neanderthals. So when you mix and you get kids, the first offspring is 50% Neanderthal and 50% modern human. But the probability that that offspring will mate again with a modern human is much greater than that it will mate with a Neanderthal because there are many more modern humans around. So over time, over generations, the Neanderthal genome ended up in us. So 70% of the Neanderthal genome is in us, and the 3% that you all carry different ones. But the medical relevance that Massimo is alluding to is very, very striking, which I think contributed to the fact that Svante got the Nobel Prize for Medicine. On chromosome 3, there is a locus, a position on the chromosome, where you can have either the modern human variant or the Neanderthal variant. That's worked by Hugo Seeberg together with Svante, not by us. And if you have the Neanderthal variant, the probability that when you get COVID-19, you will die is much, much greater than if you have the human variant. This Neanderthal variant on chromosome 3 is responsible for about 1 million dead people of the pandemic. 1 million. In other words, Neanderthals killed many more humans now than they did 40,000 years ago. There's a question.
Okay, yes, please.
Thank you so much for an incredible talk. Is RGAP11B expression inhibited at a certain stage of development? And if not, is it related to oncogenic activity?
Yeah, excellent question. Yes, it is, because RGAP11B is a cancer gene. So if a cancer persists, you will have active RGAP11B. But in normal brain development, it's turned on very early, 8 weeks of conception, something like that. And it stops around, I think, 25th week. So it goes along with the process of making neurons, and then it's shut off, luckily. If it wasn't shut off, it would be a candidate gene for terrible brain tumors like glioblastoma multiforme.
We have a question there. Thank you.
So the animal model that you use with the 11B mutation, with the 11B gene, we can say that it has the brain that is more similar to the human. So do you think that it's better to study the neurodegenerative disease in this animal model compared to the one that we use?
Sorry, can you raise your hand? Okay, yeah. So the question is, I guess you ask, how do these monkeys behave? Yes, we actually wrote that in our publication. We confined our study to the fetal stage for ethical reasons, because I expected that the brain would get bigger, but I have no idea what happens with monkeys when the brain gets bigger. Maybe they just get smarter, but maybe they get autistic or something. There are lots of possibilities there, because when they have more neurons, these neurons may not connect correctly. So we made the decision with our Japanese colleagues that we would not let these monkeys be born. We collected them by a cesarean section, 2/3 through gestation, through pregnancy. In the case of the mice, we did not have these ethical concerns, because we said, okay, if something funny happens, we can, sorry to say, but we can kill them. But it's not ethically as problematic to have an inbred mouse strain with a human-specific gene. And so far, it didn't do anything bad to the mice, no epilepsy, no nothing. They are just a bit smarter.
We have a question there.
First of all, thank you for the lecture. Hi.
Okay, I see you.
I have 2 questions, if that's okay. The first one is, do you know if there are any studies that introduce the 11B gene and also altered those 3 amino acids in the 2 checking proteins?
To introduce the gene into what?
In a primate.
Yeah, well, we did that.
No, but also change those amino acids at the same time with the checking proteins.
Oh, no, we have not done that yet. It's a great... You mean this is now not 11B, but the KNL1, KIF18A, the chromosome segregation story.
Combining both.
Yeah, we have... I mean, this is not so easy to do. I mean, when you make a transgenic monkey, you talk about 3 years of work. So we did not do both together. It's a very good point you're raising. And it certainly would be interesting to see, but again, I would confine it to the fetal stage.
Okay, and my other question is, if you believe that the genes that you spoke about could be a therapeutical target for cognitive disabilities.
Maybe not for cognitive difficulties or disabilities, but maybe for neurodegenerative diseases. So when you have a situation where pathologically neurons die, and you want to replace neurons, say in Parkinson, RGAP11B has this unique property that it can force the stem cells to first make more of themselves and then make neurons. And that ability of the protein could be therapeutically very useful. I actually submitted a hypothesis paper to a journal. I haven't heard anything yet from the journal, but I make that point that future scientists may wish to study RGAP11B as a potentially therapeutic beneficial protein.
Okay, thank you very much.
Okay, we have another question there.
Hello, I'm here. First of all, thank you for this fascinating lecture. This question is more related because I'm currently studying on anxiety in mice, and because I saw in the slide that you also, I just saw the anxiety. I just wanted to see, so in my case, my mice were almost not having anxiety because they were not that smart. So the thing is, when they become smarter, like slightly smarter in your study, do they show more anxiety? And I mean like